Hi, I'm Dominique Bergmann, and I'm a professor at Stanford University in the Department of Biology, as well as an investigator of the Howard Hughes Medical Institution. And in the second part of a three-part series on plants, development, and stem cells, I'll talk about some more detailed research on particular stem cells in plants and what they can tell us perhaps about our own. So to begin, I want to say a few ground rules about important things for stem cell biology and also some differences between plants and animals. So the first really key feature is in stem cells and in regeneration, one of the core features is the idea of an asymmetric cell division. So a division in which a mother cell creates two different daughter cells. One of those daughter cells is just like the mother, and it replenishes the mother cell, and the other one goes off and does something different. So in this diagram, we have the two different purple cells. One of these cells would act just as its mother, replenish it. That's the stem cell nature of it. The other cell will go off and differentiate. So this is something that's key for any kind of stem cell divisions that we know about. And the details can vary a little bit, but it's really a core feature. In plants, we study stem cells for a number of different reasons. One, plants are very good at generating stem cells. But the particular stem cells that I'm going to talk about, one of the prime reasons that we can study them is they're very accessible. And to bring this point home, what I want to say is that when we think about mammals and our own stem cells, these are part of the organs, and they're formed very early in the embryo, an embryo that's in utero and basically inaccessible. We can't see how stem cells are born. But on the other hand, plants are continuously making stem cells. They're making them exposed to the environment, exposed to the investigator. And so we can really capture really interesting questions about how stem cells are made, how they work, and then how they stop being stem cells. So I'm going to focus on a particular set of stem cells, and those are in the epidermis of the plant, so the skin of the plant. And those are cells that generate two different very special cell types. One is called a pavement cell, and these are these interdigitated jigsaw-like puzzle cells. Those just make a waterproof surrounding of the plant. The other cells, called stomata, are actually two cells, and those two cells work together as a valve. That valve lets carbon dioxide in from the atmosphere in exchange for water and oxygen out from the plant to the atmosphere. So together, these are all critical for the plant to live, the waterproofing to keep it mostly protected, and these holes for it to generate, to gather the nutrients, meaning the carbon dioxide that it uses for photosynthesis in exchange for water. So functionally, they're important, but what we're really focused on is how are they made. And to do this, one of the nice things is that you can watch the entire stomatal lineage, the cell types and their precursors that make these particular epidermal cells. So as I mentioned, plants are continuously generating stem cells. So we can watch the initiation, the beginning of the stem cell population. And this is an image. It's a confocal image from a microscope that has a very thin layer of the epidermis. And what you can see is a bunch of cells in a very young leaf, and they're basically the same. But some of those cells get to be different. Some of those cells, and we don't really know how, some of those cells initiate the stomatal lineage. They make an asymmetric cell division, and they begin the stem cell population. So we call it initiation. And I'm going to show you this in the same cartoons throughout this talk. So we'll see the same color coding. You'll always see green for the stem cells, and then blue and purple as they differentiate. So in the initiation, we have an asymmetric cell division. Here I've color-coded the two cells as being different. They're also different in size. Once that happens, those two cells behave a little bit differently. And I won't go through the extreme details of this, but both of those cells can divide in different ways that will actually generate one or the other cell type. Remember, this is a stem cell lineage that will generate stomata, which will always be shown in color, and pavement cells that will always be shown in white. So if you look at these two cells, you can divide in such a way that you generate more colored cells, so more stomata, or divide in such a way that you generate more pavement cells in white. And what's nice is, as I mentioned in part one, plants are developing very much in tune with their environment. If you're in a dry environment, these holes, these pores are not so useful. So you want to generate more pavement cells. In other environments, you might want to generate more pores. And so this is a flexibility built into this lineage. Now this is going to continue for a number of times. These are asymmetric divisions, and we call this amplification because this is actually building the leaf. The number of times you divide will actually generate not only two different types of cells, but cells in general. So if you divide many, many times this time, you get a very big leaf. If you don't divide so many times, you get a smaller leaf. And again, that's environmentally sensitive. So they'll do this for a while. Any one cell can generate a whole bunch of different cells. Eventually, though, these cells will stop that being in that stem cell-like state and differentiate into one or the other cell type. So those two cell types, those pavement cells again in white, and the colored cells will go through this process shown here called differentiation, where they go from blue, they divide once down the middle, and that makes those two symmetric cells of the guard cells. So one of the important things to think about is where is this happening on the leaf? Now often when we talk about stem cells, we talk about a stem cell niche, a specific area where the stem cells are born. It's a little bit different here. They're actually born throughout the leaf. So this is an image that's now colored with the stem cells in green as green circles. The committed cells, those have moved on and now are going to make stomata. Those are blue. And then the cells are just outlined in red so you can see them better. But in this leaf, what you can see is that those stem cells are everywhere. So you see green all over the leaf, whereas you see blue in a couple of other places, not quite as many at this stage. This is not the youngest leaf that I showed you in the beginning, but it's also not a mature leaf. It's somewhere in the middle of its development. So we're going to zoom in on this leaf, and I'll show you a little block in there, and we're actually going to watch a movie of that leaf develop over about a 24-hour time period. So this is that area. Again, same color coding. So the stem cells are in green. So you can see, this is actually my favorite one to watch, so watch that one. You can see some that are a little bit further along in blue. And as this movie plays, what you're going to see is it's going to look like it's moving and growing. That's actually the leaf expanding as the cells divide. So if we watch this, watch that cell divided, you can see it divide and you had two green dots flash. And then only one of those cells becomes committed. And if we watch movies like this and other cells in that movie, we can learn the rules of stem cell behavior. So whenever we see a stem cell divide, you can see this a little bit more easily in some of these still pictures. So whenever we have a stem cell dividing like down in here, we see that the two daughter cells initially have a chance to be stem cells. But if you wait an hour or so, they've either fought with one another or something has happened so that only one of them will stay a stem cell. So they're only both green for a little while. And then they're going to go off and do different things. And in this case, we have one of them committing to becoming a stomatal cell, turning blue, and the other one committing to becoming a pavement cell without any color inside. And this always happens. We can tell a little bit about how it happens. So one of the things you'll notice in the very center of this image, this is actually a mature set of guard cells. That's actually a stomata already. And that's another thing that's interesting about this system is that not everything is doing everything at the same time. So some cells have gotten all the way through the lineage and some are still stem cells. So in this case, if you already have a stoma, you don't want another one right next to it. So these cells talk to one another so that they're spaced out, and that's more efficient for photosynthesis. Okay, so we can see a little bit of the rules for here. We'd like to know what's actually controlling this. How are cells talking to one another? How do cells know that they should be a stem cell, that they should keep being a stem cell? And how do they know it's time to be done with being a stem cell? They don't need to divide any more times. They need to stop and move on to the next stage. Okay, so one of the things that's really wonderful about plants is we can make movies so you can watch things over time, but plant cells are locked next to their neighbors. And so if you watch them, you can always predict who was the sister of whom. And this is really useful if you're thinking about generation and regeneration. And I mentioned earlier, stem cells are accessible so we can see them. The other things that are useful about them is that plant cells don't die during development. So apoptosis, which is an important part of animal development, that doesn't happen here. So we don't have cells disappear, and cells don't move away. And so we have a perfect record of every cell and its division and who it was next to throughout development. So not only can you run development forwards, but you can actually, and this will come up when I talk about regeneration, you can actually see things go backwards. So this is an example of something that I think is very useful when thinking about a stem cell. So at the center of this is a bunch of cells that are related to one another. They're the products of stem cell divisions. Now currently, this bright green one is the stem cell. This one next to it was its sister. It's about to stop being a stem cell. But these two, we can tell what their sister was before, and that's the one that's right next to it here. So these two are sisters of the next largest cell, and those are sisters of the largest cell here. So we have a way of tracking who was whom throughout the whole development. And I'm going to come back to that later. You'll see how important that is. All right, so we know who the plant cells are, we know what they used to be, and we know what their potential in the future is. So what are the key questions that we want to know? So we want to know, this is something that we can really only answer in a few systems. So how are stem cells born? What makes a cell divide asymmetrically and become a stem cell instead of doing something else? Once it is a stem cell, what determines how often it divides and how many times it divides? Because these are not permanent stem cells. They are limited. They will divide only 3 to 4 times before committing to a particular fate. So if you've got cells that are doing this, though, then the next question is, well, how do you stop doing that? Once you've gone beyond being a stem cell, how do you make sure you never go back and become a stem cell again? Or maybe there's a situation in which you do want to do that, so how would you do that? And then underlying all these big questions are, what are the genes that are in charge of this? So what are the genes that are required to make stem cells, to keep them going, and to stop? And so I'm going to cover all of these in this section, focusing on stomata and those precursors that I've shown you. So again, we're going to go and look at these cells in this diagram cartooned here, but we're actually interested in those real ones that are up in the corner. So some of the key regulators, and we've known this from genetics from about 10 years ago, some of the key regulators are transcription factors, so proteins that sit in the nucleus and regulate whether other genes are turned on or turned off. And 2 of those important genes are called speechless, or here it's in green, in the abbreviation speech, or mute. Now you might be wondering why they're called this. So stoma or stomata, that's the Greek word for mouth, and that pore on the surface. And so many of the genes were named in terms of mouth. So speechless has no mouths, mute has no mouths, too many mouths, another gene has too many of them. Okay, so these are some of the key regulators. And those green and blue dots that you saw moving around, those were actually reporters telling us where speechless and mute genes are acting in the plant. Now, mute and speechless are actually very closely related transcription factors. Their sequences are very similar, and they have a sister named Fama. If Fama was in the movie, it would be showing up a little bit later. And I'm just going to show Fama in purple. And Fama is required at the end to make sure that stomata consists of 2 cells and only 2 cells. If you make only 1, it can't open and close. If you make 6, you have some problems as well. So Fama is required at the very end. Now, these can bind to DNA and activate genes or repress them only as partners. So they can't do this on their own. They all share the same partner, 2 partners called SCREAM1 and SCREAM2. And together, these are called master regulator BHLH type transcription factors. And they're called master regulators because they have a very, very big effect on development. So for example, without them, without speechless, the very beginning part of the pathway, you get no asymmetric divisions whatsoever. If you make no asymmetric divisions, you can't make any of the stomata. So you're dead, speechless. It's a very sad little plant. But that's a very powerful regulator. Now, on the other hand, if it's a master regulator, it's something that if we make too much of it, if we artificially put in too much, we should be able to convert other cells into stomata. And that's exactly what happens. So if you take mute and you make too much of that, other cells that would normally do something else are now going to make stomata. So you get a whole plant full of stomata. That's also not so healthy. So it's not dead yet, but it's not the most healthy thing in the world. Okay, so these are master regulators. They're very important for stomata. But when we discovered these, we, in our group, as well as Keiko Tori's group at the University of Washington, we kind of co-discovered these. And what was really interesting when we found them was that this idea of using transcription factors of this particular type in the particular way that they're being used looked a lot like what we knew about from animals. So both in neurons, and this is something called acute scute, and in muscles, the myod and myogenin family, these are related BHLH type master regulators. They're involved in development where you have a stem cell division in the very beginning, and you create a lot of precursors, and then you help differentiate. And so that's kind of interesting that you end up with very similar genes. I talked about in part one the fact that plants and animals, they only share a unicellular common ancestor. So everything that they do in development, they invented separately. All of the genes in the BHLHs, they've actually invented separately as well. And so both of them, even though they had all the different opportunities in the world, they both came up with the same strategy. And so maybe that tells us something very deep about how this type of gene regulation or these types of proteins are really well suited for developmental decisions like this. Okay, so those are the master regulators. Those are the things controlling it. But they're not the only genes. Plants, Arabidopsis has about 27,000 genes. Humans have about the same. They're not just one or two genes that control these things. So what we really wanted to know was not only what were the master regulators, but what were the changes? So if you have one of these green cells, what are the genes that it expresses to make that a green stem cell? What are the ones that go away when you transition into something that's differentiating? And one of the things that we were able to do, because we have these master regulators and they're expressed in those different cell types, we could make a map of development. So we can pull out the cells that are green, stem cells, and separate those from the cells that are blue and those that are purple, for example, and figure out what are the genes that are over-enriched, what are the genes that we can use as a way of saying this is a stem cell type or this is something else. So to do that, remember these are all locked together. The plants have cell walls that are keeping them all together in one tissue. There's some enzymes that can digest those away. So we have a developing plant, everything is intact. We can add, and we can separate those different cell types out. And so we do that by just adding something that digests away the walls. Then you end up with these round cells. They have a membrane around them. And all of the normal interior components of those cells. Some of them here are going to be fluorescent. The rest of them are not going to be fluorescent. And so what we would do is here I'm showing you lots of different cell types together. We would do these individually. So a plant that only has that green marker, and we're going to separate all of the green cells. And a different plant that only has the blue marker, we will separate all those blue cells. So we can do this. And then we'll just have a pure population of only those cells that are stem cells at the very moment. Now, I'm showing you a few here. You need millions of cells, and there are millions and millions of cells in the plant. If you were to look at them through a microscope, you would go blind. So luckily, there's some equipment that's been designed to do that automatically. And that's something called a fluorescence-activated cell sorter. So this is a machine that uses a laser to shine a beam at cells. So you take your cells that were in the plant, as I've shown here, you put them in something called protoplasty. That was that figure that I just showed where you have those individual cells. Some of them are going to be fluorescent, others will not. You run them through this machine, and the machine can look at millions of cells. And the machine will shine a laser and then only select the fluorescent cells into a tube, and everything else just goes into the waste. So we can then have a tube full of pure cells of one cell type or another. So once we have that pure tube of cells, we can do some things to extract the RNA that'll let us know which genes are up-regulated or down-regulated. So when we do that, and we do that for each of those individual cell types, we can get a map of the genes that are highly expressed in one cell type, but not so much in the other cell types. And so what that looks like, this is a heat map where it gives you an idea of how highly expressed a gene is. So something that's red is a gene that's highly expressed at one time, and yellow means that it's not expressed. And so this is a map. It's, again, color-coded. The gray here is something that I haven't shown you before. This is a control, a different type of cell. And so here's some genes that are not in any of our stomatal cells, but only in that other type of cells. So we're not so interested in those. But this is another panel where you have some that are just in that stem cell stage, but not at the other stages. And so by doing this, we can capture literally thousands of genes that are on at these different stages. So thousands of genes that mark stem cells, thousands of genes that mark leaving stem cells, and thousands of genes that are required for those active and differentiated and functional guard cells. So that kind of gives us an idea of what's going on. But then we also have to realize if there's 1,000 genes, that's interesting, but we probably don't want to follow 1,000 genes at once. Or it's hard to come up with knowing which of those genes are the leaders, which are the genes that are making that cell type, and which are the genes that just sort of follow along. So we'd like to know the leaders. And we had some idea of what those leaders were, because again, speechless, mute, and Fama are genes that have a very big effect, and they are transcription factors. So we thought, well, it would be very interesting to say, if there's 1,000 genes that are highly expressed when cells are stem cells, but then they go down when they stop being stem cells, how many of those are under the control of speechless? Speechless as a regulator itself. So we know that speechless gets things started. We know that speechless is on in these early cells. So what are the genes that speechless itself regulates? And we can do this with an experiment called a chromatin immunoprecipitation, or CHIP experiment. And the basic idea is that if you imagine the DNA from the entire chromosomes, there are, of course, many thousands of genes on that DNA. And these transcription factors, in order to turn on a gene or turn it off, they need to be sitting right at the regulatory regions of those genes. So if we do an experiment where we have, so green or blue, we have those proteins, and we kind of link them to the DNA, and then figure out what DNA they're attached to, we can find where across all the genes where speechless is binding. So we do that experiment and find out all the places that speechless is. And the data from that looks a bit like these graphs. So this is, on the bottom, you can see a gray line, and that's one of the genes. And these little piles, those represent how many times we found speechless associated with that part of the DNA. And so the higher the pile, the more speechless is really regulating that site. Okay, so speechless does this to a couple of genes. It does it to mute, the next gene in the pathway, and scream, which is that partner. So that was interesting. But where else? Well, it turns out that speechless binds to 9,000 genes. And in Arabidopsis, there's about 27,000 genes. So it looks like it's binding to about 1/3 of all the genes in Arabidopsis, and that's an awful lot. So we thought about that for a while, and we're still trying to work on all of the things that it regulates. But I think what it does is, there's two really key points that I want to bring up. One is that this transition from not being a stem cell to suddenly being a stem cell is accompanied by a big change in the number of genes, in thousands of genes either going up or going down, and one key regulator that's setting that. So speechless being there really makes something a stem cell. The other thing that's very interesting, and I mentioned MyoD from the animal stem cells that's involved in muscle. So MyoD and speechless are proteins that look like one another. And MyoD is the same sort of thing. So if you ask where in the human genome or mammalian genome where MyoD is found in 30,000 different places. So these master regulators that are related are recruited to a lot of different places. They probably do a lot of different things. All right, so we have something that tells us a little bit about how things get started. We have a master regulator, and we know a lot of genes get changed. Then we have to think about, how do these cells keep going? So what makes them divide 2 times or 5 times? And I mentioned before that it was very environmentally sensitive. So if you're in a condition where there's a lot of nutrients, so a lot of carbon dioxide in the atmosphere, and it's the right, it's just humid enough, you might want to make a very big leaf. And so in that case, you want to make the stem cells need to divide a lot of times. So how does that work? Well, it turns out it's related to how speechless behaves, and I won't actually talk about this in detail, but work from a number of other labs have found that speechless is a target. So not only does speechless regulate many things, but speechless is regulated by many things. So how much light you have and how much carbon dioxide, there are things that sense that, and actually the readout of the environment is on how stable speechless is. If speechless is very stable, stem cells divide a lot of times. If it's very unstable and goes away, then that's the end of the lineage. So when you think about this lineage, we have a massive reprogramming from what things were before. Lots of genes change, and speechless regulates those to create this stem cell lineage. Then that stem cell has got to be flexible, and I mentioned the environment is feeding into that. But then we get to the end, and here's this issue. How do you commit? So this is all about how you get things started with speechless, but at some point, speechless is gone, and cells, they're only functional if they stay that cell type. So if that valve started to think that it wanted to become a vascular cell, it's not going to work anymore. So how do we make sure that cells stay the cell type that they should be? So this is another story, and this is a story centered on Fama. Again, remember, Fama is another BHLH-type transcription factor. It's related to speechless. But Fama does something different. And again, this is something where we see animals and plants doing a very similar thing. So Fama works in combination with a protein called retinoblastoma. And retinoblastoma has been studied by a lot of people in terms of tumors. And this is an example of the phenotype of a human who has this eye tumor. It's mostly affecting small children. And what happens is the eye overproliferates in the retina. What's interesting is, although this was a tumor that was first found in the eye, retinoblastoma as a protein is actually in every cell of your body, and it's controlling how much those cells divide. So retinoblastoma does a lot of things, but it's normally a tumor suppressor. It normally keeps cells from dividing, and it does that by holding on to some of the proteins that would normally make the cells divide. It also can do things where it locks cells in a permanent state. So retinoblastoma as a protein can link to other proteins that then shut down genes permanently, or turn them on permanently, but mostly shut them down permanently. So what is this doing in a plant, and how is it related to Fama? So what we found, actually, is that there is a particular binding site on retinoblastoma and Fama that match one another. And so they physically form a complex. And somehow, and this kind of makes sense, retinoblastoma is a protein that needs to regulate DNA, but it can't necessarily find it itself. And so it piggybacks on other proteins like Fama that can find DNA and bind to it. So the two of them go together. So these guys bind, and that normally makes them work. But what we wanted to know was what would happen if we broke that bind. Now we know that a retinoblastoma, or the plant homologue called retinoblastoma related, or RBR, that's actually in every cell in the plant. And I showed you that Fama is in a very specialized cell. So we know that retinoblastoma does many things without Fama. Okay, so what we're interested in doing is seeing if we can make a version of Fama that can't bind to RBR anymore. So they can do all the things that they normally do, but they can't interact with one another. So we're going to look like that. It's going to have this different shape. And it's important that they do their normal jobs. So one of the things that we wanted to know is if we have a version of Fama that can't bind to RBR anymore, what does the plant look like? So if we substitute the normal Fama, we get rid of the normal one and put this other one in, do we get a plant that's alive? And we do. We get a plant that's a little bit smaller than normal, but it makes leaves and flowers and roots. So we haven't screwed up too much. But again, we're interested in what happens at the end of this pathway. And so remember that I color-coded everything in purple, and stomata are usually two cells and a hole in the center. So when we don't have Fama and RBR able to interact with one another, this is what happens. So this is an image looking at the surface, and you can see a stomata. You can see two cells in a hole, but I think you can also see that there's stomata inside of stomata. And there are more extreme things where we have stomata inside of stomata, inside of stomata, inside of more stomata. And this is not normal. But so something is being reset. And one of the nice things, again, about plants, the fact that plant cells are locked next to their neighbors, is that we can see this. We can see not that just something went wrong and screwed up, but we can see that we got to the end of a developmental pathway, something that should normally stop, but these cells kept going, and they actually repeated what they did before. And we know that they repeated what they did before. They went back to that stem cell state, because we can look at whether or not those early markers, those green and blue, mute and speechless, that are normally turned off, if those come back on, And in fact, they do. And so when you make extra divisions and they start to reset themselves, what's happening is that these cells, at the end, the stomata don't remember that they should be stomata anymore, and they actually go back to the beginning. Now, one of the questions, though, is where do they go back to? So I mentioned that this was a stem cell lineage, the meristemoid. It's specialized in the leaf. And so that's the example across the top here. But those cells came from the shoot meristem that's making all of the leaves. And those cells came from an embryo, and those came from a zygote. And so when we repeat something, just how far back are we going? And it turns out by looking at whether or not the genes that get turned on in the stomatal lineage versus genes in the embryo and the zygote, it turns out that when we have this problem of going back, it actually only goes back to the speechless stage. So it's been reset, but it doesn't reset back to 0. It just resets back to the beginning of that stomatal lineage, that stomatal stem cell stage. So that's really interesting. So we have these two factors. Remember, speechless and Fama are sisters. They're very closely related proteins. So they have a lot of the same properties. We have one that massively starts the lineage. And so what can we conclude about this is that we've got something like speechless that opens the door to a new lineage, resets it. And then you have something related to it that shuts the door at the end to make sure that things are locked in place. And thinking about that in terms of what we know about from animals, and the fact that they're using proteins that we know about from animals, this is really interesting. We've noticed for many years that the same types of proteins get used over and over again in different contexts. We've always wondered why in muscles and in neurons, why do you use proteins that are closely related to one another in series in these sets? And one idea that's coming from this plant study is that you might want things as bookends. When you make a stem cell lineage, you have one factor that can bind to and activate genes. That you might want to have things started, but you might want to have its relative shutting the door at the end. So I think, in general, these are specific details about specific genes involved in a specific type of development in plants. But I think because of those commonalities between plants and animals, there are things that we ought to be looking at animals. Now, why didn't we find them in animals first? Part of that is just because it's hard to see it. And so by using things like plants or planaria or regenerating limbs or a lot of different organisms out there in nature, we can actually get some idea about how things are done and then test them in other things that we care about. And so this is my favorite little organism here, this Arabidopsis, but I think it's very powerful in telling us about some other things that we do care about. So this is work that was all done by members of my lab, and I have a really, really wonderful lab group. This is some of them, not all of them, but I wanted to highlight some of the contributions from people in specific projects that I've talked about. So this last project where I talked about Fama and retinoblastoma that were shutting down a lineage, that was work done by Juliana, Ansun, and Charles. They worked together on that. The beginning of the lineage was also Ansun, so he does double duty, but he was working there with Kelly and Cora. And Kyoko actually started this project a number of years ago. And Jessica Chang is our computational biologist who has her hand in everything that we do. So those are really key members. And that map that I showed you of all the different stages, that was spearheaded by Jessica Adrian. So they've been working together on this. We've been funded by a number of really wonderful resources. The Howard Hughes Medical Institution and the Gordon Betty Moore Foundation have really made an investment in plant science, not just me, but 13 other people were recently put together as Howard Hughes and Gordon and Betty Moore investigators, and that's really, really been key. We've also had support from the National Institutes of Health and the National Science Foundation.